Welcome back to the third lecture, and we're going to be talking about staining techniques. Our first technique is known as a simple stain. A simple stain is the usage of a simple dye, one single dye, not simple, a single dye, to stain a specimen. There are two types of dyes that we use in staining. We have basic dyes, which are positively charged. And basic dyes are attracted to the negatively charged cells of the bacteria. And so in biology and chemistry, opposites are attracted to each other. Think about magnets. And so we have the negatively charged cell, and then we have this positive basic dye. The dye is going to adhere to the cell itself. And so that's what you're seeing in this example. We have a cell, negatively charged. We add basic dye. It's going to stain the cell. The background is going to be clear. If we want to stain the background instead, then we use an acidic dye. Acidic dyes are used in negative staining. The example of an acidic dye that I always like to use is called nigrosum. It's a very easily used dye. But in this stain technique, we don't fix our cells first. So we're actually, we tend to use negative stains on cells that don't fix well or that don't take in stains well. So with acidic dyes, they are negatively charged. So is the cell, and therefore they're repelled from each other. And so instead of staining the cell, we stain the background. And so the cell itself is a clear spot on a colorful background. So we can use differential stains. Well, I will quickly mention that with simple staining techniques, the only thing we're going to learn about our cells is what is the structure of the cell and how big is the cell. That's all you learn. The color doesn't mean anything because we added the color to the cell. In a differential staining technique, we can actually learn a little bit about the cell itself, like what is bound within that cell wall. And in this case, the color actually reflects the difference in the structure of the cell. So the first and the most commonly used staining technique is called the gram stain. Sorry, hold on. Those were two of my dogs. Apologies. The gram stain is the most commonly used stain technique that we use. It separates bacteria into two very large groups known as gram positive and gram negative. And the difference is associated with what is found within their cell wall, which we'll talk about in a little bit. So how does this work? We first fix the bacteria to the slide. Then we add the primary basic stain known as crystal violet. Crystal violet stains all of the cells the same color. We then rinse the slide with, after 1 minute, we rinse the slide with distilled water, and then we add iodine, which is the mordant. So mordant is kind of like a glue. This is going to produce a crystal violet iodine complex that is found within the layers of the cell wall. Once that's set for about 1 minute, we again rinse with distilled water. We then add a decolorizer. The decolorizer is acetone, and this is where we're going to see a difference. Any cells that are gram negative have very little peptidoglycan in their cell wall, and so they're going to decolorize. So they're going to turn clear again. But the cells that are gram-positive retain that purple color. We again rinse the slide with distilled water, and then we counter stain. So use our second stain called safranin, which is a light pink color. Safranin colors the cells, but it's only going to color the cells that are not already colored with purple. The purple is so dark that the pink would not be able to affect the color. And so when we're done, we rinse the slide and that with distilled water, blot it with bibulous paper, and then look at it under our microscope on the oil immersion. And we'll see gram-positive purple cells and gram-negative pink cells. So the success of the grand stain depends on multiple multiple things. So one function or one thing that I've learned that people struggle with is leaving stains on for too little time or over decolorizing. So adding the decolorizer but leaving it on for too long. Or using cultures that are very old. So there are certain bacteria that lose their ability to retain the gram, the primary stain. And if they lose their ability to retain the primary stain, they might be gram-positive, but they might show some pink cells. And so if you look here, you can see light purple and a more dark purplish color. The difference is the cells are older or they have over decolorized. But you can also see those negative cells like these that are obvious negatives. The next stain technique is an acid fast. This is also a differential stain. This stain technique is used to detect organisms in the genus Mycobacterium. So it's not as highly used. You're not going to use an acid fast stain to see if you have a staph infection. It would not work. It would only tell you that you don't have Mycobacterium. But Mycobacterium contain certain organisms that cause some very bad diseases, including tuberculosis and leprosy. And for that reason, the stain technique is still very important. The cell walls of Mycobacterium contain a waxy substance known as mycolic acid. And because of the mycolic acid, they don't stain very effectively. And so we use a procedure known as the acid fast stain to allow the cells to take in the acidic dye known as carbonyl fusion. So carbonyl fusion will bind to the mycolic acid and will stick to that mycolic acid. It does not bind to other structures. Because of that, it will stain the acid fast positive cells that contain mycolic acid with a pink color. We then decolorize using acid alcohol. Anything that is not acid fast is going to become completely clear or colorless, and we counterstain with methylene blue. And so on here you can see a few acid fast cells, and you see a lot of blue cells. This stain technique actually takes about 15 minutes in the lab because we're doing it over a steam plate. So you're steaming that initial carbonyl fusion into the cell. A capsule stain is very much like a negative stain. So with the capsule stain, we start out with a negative stain that colors the background. We then, once the slide is dry, we then add safranin to the cell or to the slide, and then we blot with bibulous paper. And when you do that, you end up with these cells that have a pinkish color, and you have a background that is dark. And surrounding that surrounding the pinkish cells are the capsules, which have no color whatsoever. The endospore stain, this is another special stain that allows us to identify cells that persist in the environment, known as endospores. So endospores don't stain very effectively, but the cells themselves will stain effectively. And so if you're doing a gram stain, you will see these pink cells. Oftentimes the pink cells have clear spots inside of them. That's because they have endospores, because endospores don't really stain effectively. So with an endospore stain, we're going to steam, again, the slide with the initial stain. The initial stain is known as malachite green. Malachite green stains endospores, and it will sit on the slide for approximately 15 minutes while it's steaming to get the malachite green to set into those endospores. Once that happens, then you rinse with distilled water and counterstain with safranin for another minute. When you're done, you're going to end up having the vegetative cells, the bacillus cells, and you'll have endospores as well. The bacillus cells will be pink, the endospores will be this greenish color. Two genuses that produce endospores are Bacillus and Clostridium. Clostridium are anaerobic, Bacillus are facultative anaerobes. So they can grow in both oxygen or non-oxygen environments. They prefer oxygen though. A couple stains that we do not do in our lab, we do not do the Flagella stain, but it is an effective stain to allow you to see the flagella of bacteria. If you see flagella, then you know that the bacteria are able to be, or are considered motile. They can move around. If there are no flagella, then you do not have motile species. And so over here you can see the flagella on the edge of the cells. Here you can see structures flagella as well. And the last stain technique is fluorescent stain. We, again, don't use this in our labs, but it is used in hospital laboratories in the immunofluorescence assays that they do to identify antibodies in individuals, or I should say, or to identify certain types of pathogens in individuals. All right, I'm going to stop here. It's been almost a 15-minute video, and we'll get into part 3, or section 3.3, which talks about cell structures, okay? Bye.